So what happens when this action potential hits the end? So we saw how it propagates, right? So sodium channels are going to open, it's going to depolarize the membrane in that spot, but that depolarization is going to leak to the next set of sodium channels, and then they're going to open, and it keeps going down that way. It keeps going along, going along. Here we can see number one is referring to the action potential coming down. So as this action potential travels down the axon, it's going to keep traveling the same way we just talked about, sodium channels opening and bringing positive charges in that kind of go down to the next set. But when it reaches the end, one of the major things that happens is it's going to open another set of voltage gated channels, but these are calcium channels. So voltage gated calcium channels. Now calcium also is higher outside than in. And so what ends up happening here is calcium comes flooding in. Now calcium is different because calcium is able to help trigger the exocytosis process. Now, what is going through exocytosis? Well, it turns out that there are these little vesicles. Remember vesicles? Remember when we talked about exocytosis? Vesicles are these little like membrane-bound things that have something inside. Well, in a neuron, there are so-called synaptic vesicles. These are things that contain a set of neurotransmitters. We've talked about these too, although we haven't really talked about what they do yet. Neurotransmitters, remember, are chemical signals that are going to transmit a signal from one neuron to a target cell. In this case, the target cell, they're just calling a postsynaptic cell. It looks like probably another neuron. Doesn't have to be, it could be a muscle cell, it could be a gland. There's different kinds of cells that it can be here. But in this case, we're probably talking about another neuron because neurons talk to each other a whole lot. So calcium comes flooding in. It's a voltage-gated calcium channel, comes flooding in. It triggers the synaptic vesicle to fuse with the membrane. That's exocytosis. So this is the exact exocytosis process that we've talked about before. But in this case, it's these synaptic vesicles that are full of neurotransmitter. The neurotransmitter will cross the gap. Now, these cells are very close together. And so it doesn't have to go very far. And there's not a lot of space in here, so you don't even have to have a whole lot of them to get a pretty high concentration in this space. So by having a really small gap, you don't need a lot of neurotransmitter, but you need enough. So the neurotransmitter will diffuse across this synaptic cleft. Again, if you're waiting for the speed of diffusion, we know diffusion doesn't happen super fast. So the fact that this space is really short means that this can happen quickly. It then binds with receptors on the postsynaptic cell. And when that happens, there's going to be a response on the inside. One response, let's talk about responses. I think that's on the next slide. Nope. I might have to come back to that. All right, so I think there's a slide that talks about that, but it's not quite there. One response that can happen is these become ligand-gated ion channels. So not voltage-gated, but ligand-gated, where the neurotransmitter is the ligand, binds to this channel, opens the channel, and ions come flooding in. And so in this case, when the ions come flooding in, it's going to give a graded potential, not an action potential, but a graded potential here. And that goes back to where we started before. So we talked about how graded potentials can sum up to maybe trigger another action potential. And so these are going to start the graded potential. The ions that are let in or out are going to decide whether this is excitatory or inhibitory at this point. Okay. So if you get sodium or calcium coming in, that's going to make it less negative on the inside. Those are going to be excitatory. If you have chloride channels, so chloride coming in, that makes it more negative on the inside. That makes it inhibitory. Or calcium going out. Like if anything you can imagine, it has to do with what it does to the membrane potential. Another possibility is that these neurotransmitters can trigger a second messenger cascade, kind of like a G protein thing that we've talked about before. And that is also a possibility here. Once the signal is transmitted, this whole system needs to reset. And so the neurotransmitter that got released has to get out of there. You have to basically remove it back out again to get the concentration low enough that the signal turns off. And so there's three ways shown here. One is just reabsorb it. And so basically have some kind of a transport protein that is going to bring the neurotransmitter back inside. And so here you can see it coming back into the axon, which is a great way to do that, right? Bring it back into the axon terminal here, so that it could just be repackaged into another synaptic vesicle and recycled and used again, right? That's a great way to do it. Some glial cells, other neural support cells might also pick that up. Also a possibility is to have enzymes in the cleft here that immediately start breaking down the neurotransmitter. So as it's being released, the opening up of the vesicles through exocytosis releases a whole bunch of them out, but then these enzymes start breaking them down right away. And so the concentration comes out high really quick when they all get released out, but then very quickly goes down again. So you get this like quick response, but then it goes away. The third thing is simply that the neurotransmitters might diffuse out of the cleft and away from the receptors. Synaptic fatigue can occur when a neuron gets stimulated over and over and over and over and over again at a rapid rate. It can lose the ability to fire signals, and one of the reasons for that is it can basically use up all of its neurotransmitters here. It releases them all out and doesn't have enough to keep the signal going. Yeah, that's the main thing that can happen there. pH also can influence neuronal activity. Alkalosis can increase activity. Acidosis can decrease activity. Neuron excitability is dependent on oxygen supply. It takes energy to be able to maintain gradients and to be able to build and maintain vesicles and to do exocytosis. So oxygen is energy. And then different drugs can increase or decrease the excitability of neurons by a lot of times by affecting the neurotransmitter receptors on the membrane, or possibly the release here. One of the things we talk about drugs increasing or decreasing, a lot of times this happens because the drugs can either mimic or block different neurotransmitters. So let's talk about some of these different neurotransmitters and what they are. A lot of these are important. These are just different categories. One of them by itself is acetylcholine. We're going to hear about this one quite a bit. This is synthesized from a compound called choline. And acetyl coenzyme A puts the acetyl group here onto a choline molecule. These are found a lot by cholinergic neurons. That's just what cholinergic means. It means it uses acetylcholine. And central nervous system, the autonomic nervous system, and the somatic motor system. So somatic motor means like your voluntary muscles. Autonomic is referring to involuntary. We'll talk about those in an upcoming lecture. Amine neurotransmitters are derived from single amino acids. So serotonin, histamine, dopamine, norepinephrine, epinephrine are all pretty closely related. These are derived from a single amino acid. They are not amino acids themselves. They are primarily used by neurons in the central nervous system, although norepinephrine is also used in the sympathetic division of the autonomic nervous system. That's the fight or flight version of that. So we'll talk about that one as well. Different amino acids can be used as neurotransmitters. So these are often used in the central nervous system. Glutamate and aspartate are two examples. These are part of the 20 amino acids that make proteins as well. Glutamate, also known as glutamic acid. You might have heard of monosodium glutamate, which is MSG, often used to make food a little bit tastier. but some people have headaches and other neurological responses, maybe in part because of its use as a neurotransmitter. These are both excitatory, which means they are going to take their target cell closer to threshold and make it more likely to do what it's going to do, have an action potential. GABA, which is gamma aminobutyric acid, and glycine are two inhibitory neurotransmitters, which means they are going to hyperpolarize their target cell and result in that target cell being less likely to fire. Another category are neuropeptides. These are made from chains of amino acids. They behave differently in that small quantities of them can have very long, prolonged effects. They are bigger, they are harder to diffuse away. Examples include oxytocin, which I know we think of as a hormone, but remember, just like we talked about norepinephrine on the previous slide, the difference between a hormone and a neurotransmitter has only to do with how it's released. The same chemical could be either one, right? So oxytocin, if it's released in a synaptic cleft, would be a neurotransmitter. If it's released into the bloodstream, would be a hormone. Substance P, and then opioids, like beta endorphin, shown here is an opioid receptor. Purines, these are nucleotides that function as neurotransmitters, so ATP and adenosine can actually act as neurotransmitters, as well as, of course, their other functions. Nitric oxide is a gas that can function as a neurotransmitter. It gets produced and secreted as needed because it's a gas. It can just float through, basically, the cell membrane. So it just goes across the gap, just diffuses across the membranes of both cells. It's a very small molecule, can diffuse pretty quickly. And so it just will cross and bind to proteins within the target cell. So it actually is getting made as when it's used or needed, as opposed to the other ones, which are pre-made and packaged in those vesicles and waiting. And then there are some lipids that can serve as neurotransmitters. They bind to the cannabinoid receptors as part of the endocannabinoid system. Endocannabinoids are basically the system that cannabis, basically marijuana, THC, what those chemicals act on, they act upon that endocannabinoid system. So we have, if you think about a drug, the way drugs work is they are interacting with something that's already in our body. And so the endocannabinoids, endo meaning from like internal, intrinsic to us, they are natural, normal cannabinoids, whereas THC would be something coming in from outside as somebody is using marijuana. Basic summary of these different groups, or at least of some important ones. So acetylcholine, we're talking about skeletal muscle contraction, as well as signaling in the central nervous system and in the peripheral nervous system. Glutamate is the main excitatory neurotransmitter in the brain. GABA is the main inhibitory neurotransmitter in the brain. Glycine is the main inhibitory neurotransmitter in the spinal cord. Dopamine has multiple things, but it's best known for being involved in reward and motivated behavior pathways. A lot of times it's associated with addiction. Also movement. Parkinson's, for example, is a deficiency in dopamine, dopamine-releasing neurons. So we might talk about that later. Serotonin has a major role in mood. A lot of antidepressants are serotonin reuptake inhibitors, which means that as serotonin gets released, we have these transport proteins that take it back out, and antidepressants basically help it stay there. So help the serotonin stay in the synaptic cleft and not get taken back up as fast. And so that helps apparently fight depression. Also cognition and learning. Norepinephrine helps to increase alertness and fight or flight. Okay, I'm going to pause here and finish up next one.